The macrophage is activated when there is some kind of infection, when some pathogen is in the surrounding or comes in contact with the macrophage, then the macrophage is activated. So, as I told you, pathogens that are extracellular or pathogens that are intracellular are ultimately leading to the production of different kind of cytokines by the macrophage. Okay? So, at the site of infection, activated resident macrophages secrete inflammatory cytokines. And what are these cytokines that the macrophage is going to release? There are different kinds of cytokines. Some of them are pro-inflammatory cytokines that are going to enhance the process of inflammation. Some of them are anti-inflammatory cytokines that are needed to stop the process of inflammation. It depends upon the situation. So when the macrophage is activated as a result of some interaction with the pathogen, then it is going to secrete these very important cytokines. One is interleukin-1 beta. The macrophage is filled with interleukin-1 beta. Already there is a lot of preformed interleukin-1 beta. It's very interesting that how the interleukin-1 beta is going to exit the macrophage or come out of the macrophage. And there are other cytokines like the tumor necrosis factor alpha, interleukin-6, CXCL8, CCL2, and interleukin-12. So these are the most important inflammatory cytokines that would be secreted by an activated macrophage. Okay. What is the tumor necrosis factor alpha going to do when the macrophage is going to secrete it? It induces blood vessels to be more permeable. We talked about the blood vessels permeability, that how the blood vessel is going to give way to the cells of the immune system and products of the immune system to come out of the vessels in the surrounding tissues, or enabling cells, fluids, and soluble effectors to enter infected tissues. And the permeability is increased, the vessels are dilated, then migration or transportation of these cells or leakage of these cells from the vessels into the tissues would become very easy. So that job is accomplished with the help of tumor necrosis factor alpha. Then there is interleukin-6 secreted by the macrophage, and it induces fat and muscle cells to metabolize, to start metabolism, and generate heat and raise the temperature in the infected tissues. You see that when there is inflammation, the temperature is increased. The temperature of the inflamed area is increased, and it feels hotter as compared to the rest of the body. So that is achieved with the help of interleukin-6, which is going to increase the metabolic rate in that particular area where the macrophages have secreted interleukin-6. And that area, of course, is the area where there is pathogen, where the infection has just started. Okay? So the inflamed area is hot because of increased metabolism due to interleukin-6. And then there is this attractant or chemokine factor, CXCL8. It recruits neutrophils from the blood and directs them to the infected tissues. Who is going to invite the neutrophils? The macrophage is going to do that. And the macrophage is doing that with the help of a specific chemokine or chemoattractant cytokine, which is CXCL8. It is going to make a gradient of this chemokine right from the infected tissues to where the source of the neutrophils is, and that is the blood circulation. The permeability of the vessel has been increased, the chemokine gradient has been built, so the neutrophils are going to follow that chemokine gradient from the blood vessels to the site of infection. Okay, so why the neutrophils are needed at the site of inflammation? Because just like the macrophages, neutrophils are also phagocytes, and they are going to engulf the pathogens that are present at the site of infections. They are going to help with macrophages to clear the pathogens from our bodies. That's why they are needed, and they are going to secrete more cytokines and to enhance this process of clearing of the pathogens with the help of other cells, and also by contributing themselves. So this is about the CXCL8. Another chemokine, chemoattractant cytokine is CCL2, and this recruits monocytes from the blood and directs them to the infected tissues. About the infected tissues, tissue-resident macrophages. That the macrophages are present there inside the tissues. We call them resident macrophages right from the birth, when the baby is born. But even before that, the macrophages have their origin that can be traced back to the yolk sac when the embryo is being formed. So the tissue resident macrophages, actually, they have their origin from they have an embryonic origin. They just have come out of, I don't know, people have started calling me. Okay. So those are the tissue-resident macrophages. But monocytes, which are inside the blood circulation, when these monocytes are, transported from the blood into the tissues, they also become macrophages. We call them macrophages. So monocytes are circulating macrophages. They are in the circulation, blood circulation, but in the tissues they are macrophages. So sometimes when there are some infections, in those cases, the population of the tissue-resident macrophages but only they can live longer lives only when they have not come in contact with the pathogens. When they come in contact with the pathogens, then the life of the macrophage, just like the neutrophil, is finished. The neutrophil is not going to survive longer once it has come in contact with the pathogen. Why? I would also tell you about that. Okay, so this population of the tissue-resident permanent macrophages can also be reduced as a result of infections in the tissues, and there may be, the need for, reinforcement. So reinforcement is in the form of monocytes. That monocytes should go from the blood circulation into the tissues to compensate for the loss that already has happened at the site of infection in the form of losing macrophages there, okay, due to infection. So who is doing that? The CCL2 chemokine, chemoattractant cytokine, which the activated macrophage is secreting at the site of infection, okay? So on one hand, they are inviting neutrophils. On the other hand, they are also inviting monocytes to enhance this inflammatory process further. Then there is another cytokine, secreted by the macrophage, which is interleukin 12. And interleukin 12 recruits and activates natural killer cells to secrete cytokines that strengthen macrophages' response to infection. Okay? The natural killer cells, you know about the function that the natural killer cells do. Okay? The natural killer cells are naturally equipped with this ability to kill the infected cells. Many of the pathogens, when they are infected with some microbes that live inside the cells, it becomes harder for to just clear that microbe from your system. But the natural killer cells, they know about the cells. They have this capability that this particular cell is infected and it should be killed. And another thing is that also these natural killer cells are going to secrete certain cytokines that are going to help with macrophages and increase or enhance the response of the macrophages to secrete more pro-inflammatory cytokines, just like these one, okay? To secrete more interleukin-1 beta, to secrete more of the tumor necrosis factor and other chemokines, and just further this process of inflammation. So this induces a very potent inflammatory response in the tissues. And when all these cells arrive at the site of infection, then these cells would just, clear the microbe from the system. So that's how the activated macrophage actually would behave. And if you look at, interleukins that are secreted there in the interleukin-1 receptor signaling. Interleukin-1 receptor, this is very important because interleukin-1 receptors and the toll-like receptors that we were discussing in the previous lecture, they have the same TIR domain, the internal domain, which is responsible for signaling. When the receptor comes in contact with the pathogen, whether this is a toll-like receptor or interleukin-R receptors, is they have the same TIR domain. They are going to pass the signal the same way, in the same manner. Okay? So this kind of signaling is central to the innate immunity and inflammation, because the innate immune response in the form of this inflammation, which is the goal of the innate immune system or the innate immune response to induce the state of inflammation, is not possible without interleukin-1R or the toll-like receptors and their signaling. Okay? So what is interleukin-1? Interleukin-1 is a cytokine, and it's not a single cytokine. Interleukin-1 is a whole family of cytokines. There are 11 different cytokines in the interleukin-1 family. But the most important one is interleukin-1 beta. Okay? This is considered as the most important because in most of the studies, they have implicated that interleukin-1 beta is involved in chronic inflammation. This is called the master regulator of inflammation. Several studies have been conducted where people have used antibodies against different cytokines, and they have observed that if you use, for instance, a monoclonal antibody against tumor necrosis factor alpha that you study, okay, or different chemokines that the macrophage is secreting, or interleukin-12, all these 5 different cytokines that you just saw there. And if you could raise monoclonal antibodies against these, and you use these monoclonal antibodies as therapeutic agents to subside the inflammatory process, just like for instance, people that are using NSAIDs or the non-steroidal anti-inflammatory drugs or the steroidal anti-inflammatory drugs in order to relieve the pain or to suppress the process of inflammation. Similarly, you can also use monoclonal antibodies to target different cytokines and subside the inflammatory process, So if you have monoclonal antibodies against these 5 different cytokines, and you design an experiment in order to see that what kind of cytokines actually can, what kind of monoclonal antibodies are interleukin 1 beta monoclonal antibody or anti tumor necrosis factor alpha antibodies or anti-interleukin 12 antibodies, which type of antibody is going to be more effective in suppression of the inflammation, then it would be interleukin 1 beta. Many researchers, they have come out with this conclusion. They have conducted different experiments that this is a very potent, uh, regulator of the inflammation, because when you use monoclonal antibodies to suppress interleukin 1 beta, the inflammatory process is significantly reduced or stopped. Okay? And if you use monoclonal antibodies against other cytokines, the inflammatory process is not, you can say it doesn't stop with that intensity as you would see in the case of interleukin 1 beta. So that's why we call it the master regulator of the inflammatory process. This is synthesized in the macrophages and other cells, and it is synthesized on the ribosomes. And as I told you that, the macrophage is going to synthesize large amounts of interleukin 1 beta. Okay, so there are large reserves inside the macrophages, and these reserves are in the form of this 35 kilodalton molecule, which is not active. This is inactive interleukin 1 beta that can also be called as pro-interleukin 1 beta. Okay, so that pro-interleukin 1 beta or the inactive form of the interleukin 1 beta 35 kDa molecule is of no use. This is inside the macrophage. Why such large amounts are synthesized inside the macrophage? Because macrophage is the main cell which is going to start with the process of inflammation. It has to set the alarm for this process of inflammation. It is going to invite all other cells, as you saw. That's why it is not possible without the large-scale secretion of the interleukin 1 beta. So interleukin 1 beta is already there because it is needed for the macrophage's immediate response in situations when there is some infection or the pathogen comes around it. So how this molecule is going to be activated? This activation actually is accomplished with the help of a special structure which is made inside the cell, and that structure is known as the inflammasome. Inflammasome is going to activate this particular interleukin 1 beta. What is there? There are different peptides coming together in the inflammasome, and one of those peptides is the procaspase 1. So procaspase 1 is the inactive form of the enzyme which is known as caspase 1. The caspase 1 is going to cleave this particular 35 kDa molecule. The procaspase 1 first is self-activated. It activates itself in the inflammasome, that structure which is made of different peptides. And then when caspase 1 is activated, it is going to cleave this 35 kDa molecule into 2 pieces. And one piece which is about 17 kilodalton, is the active form of interleukin-1 beta. Okay? So this active form of interleukin-1 beta is going to come out of the macrophages. There should be a mechanism for it to come out because this has been activated inside the cell or inside in the cytoplasm, you know. Now how it is going to come out, that would be discussed in the next slide. But once the macrophage is activated and it secretes lots of interleukin-1 beta via a feedback loop, there is a feedback loop which is established. When the inflammasome would just, help with the activation of the interleukin-1 beta, and then it just comes out of the macrophage. This interleukin-1 beta, it can act upon the same macrophage, or it can act upon a neighboring macrophage, or this interleukin-1 beta can also affect other cells that have interleukin-1 receptors. Because interleukin-1 beta, it binds with interleukin-1 receptors on any cells where there would be the interleukin-1 receptor, okay? And thus increasing the production of this particular cytokine. So this interleukin-1 beta, it maintains activation of the infected cells via autocrine or paracrine mechanism. Autocrine means that the same cell is making interleukin-1 beta, and the same interleukin-1 beta is acting upon the same cell again, okay? Because it has got these receptors where the interleukin-1 beta can bind. The paracrine mechanism is that one cell is making interleukin-1 beta, and when it comes out of that cell, it is going to act upon another cell, in the surrounding. So usually the cytokines, they are short-acting molecules. They are not long-acting molecules. And they can also do a lot of damage if they are just secreted openly into the surrounding of the cells. Huh? Aistable. Okay. Autocrine, when the same cell is making interleukin-1 beta, it is the same cell, that makes interleukin-1 beta, and the same interleukin-1 beta is acting upon it after that, when it is secreted. And the paracrine is that one cell is making interleukin-1 beta, secreted, and it acts upon some other cell. And that's why I told you that this, the cytokine secretion and the effect of cytokines on other cells, for that, because it can lead to the destruction of other cells, or it can lead to, inducing some changes in many different types of cells. That's why this paracrine mechanism in the case of cytokine is very tightly regulated. Some of the cells, when they have to secrete some cytokines, then they would go there and first make a contact with the cell and then secrete cytokines in that surrounding. And so because these cytokines would not act distantly or in distant places.